All right, today we're going to talk about something huge, something that's literally decoding the blueprint of life. It's this one concept that connects everything from cancer research all the way to, well, figuring out our own ancient family tree. But where does this high-tech idea actually pop up in our lives? Let's start with a question that I think was on everyone's mind not too long ago.
Yeah, think about it, right? How on earth were the COVID-19 vaccines developed in record time? It felt like science just hit the fast-forward button. But what was the secret sauce? What was that one key that unlocked the whole thing?
Well, it turns out the answer, it's a fundamental scientific tool. It sounds super complicated, but the idea behind it is actually beautiful in its simplicity. It's called sequence alignment, and you can think of it as the starting pistol that kicked off that whole race for a vaccine.
Okay, so a pandemic-stopping tool? That's a big claim. So let's zoom out for a second. What makes this one technique so unbelievably important? Because, as we're about to see, its impact goes way, way beyond just this one virus. It's literally reshaping, well, pretty much all of biology. I mean, just look at this list. The scope here is just wow. For the COVID vaccines, right? Scientists aligned the virus's genome to find that all-important spike protein. In the fight against cancer, it's used to compare normal genes with mutated ones to find the exact culprit. It lets us design new drugs, trace our family tree back millions of years, and even make a really good guess about what a brand new, never-before-seen protein might do, all from this one idea.
So, we've covered the why, and it's a pretty epic why. So now, let's get into the what. What is sequence alignment? How do we decode life's language?
Okay, so the official definition sounds a bit academic, right? But here's the deal. At its heart, it's just a method for arranging those long strings of DNA, RNA, or protein, the A's, T's, C's, and G's, to find spots where they're similar. Think of those similar spots as clues. They tell you, hey, these two things might have a similar function or a similar structure, or maybe, just maybe, they share a common ancestor. It's how we turn a giant mess of data into actual usable knowledge. Probably the best way to get your head around this is to forget about biology for a second and think about sentences. Look at these two. They're pretty similar, right? The quick brown fox versus the fast brown fox. That's a mismatch. Quick and fast means something similar, but they're different words. Then you have jumps versus jumped. Kind of like an insertion or deletion, what we call a gap. Sequence alignment is just the computerized process of lining these things up to spot all those matches, mismatches, and gaps in the most logical way. Okay, so we get the goal, line stuff up. But how do scientists actually do it? Turns out there are two main ways to go about it. Two fundamental approaches. So the two big flavors here are pairwise alignment and multiple sequence alignment. And the names kind of give it away, right? Pairwise is just that, a pair. You're comparing two sequences head-to-head. Super useful for finding a direct relationship. But then you have multiple sequence alignment, or MSA for short. This is the big one. It aligns 3 or 4 or 100 sequences all at once. This is what you use when you want to look at entire families of proteins or build one of those big branching evolutionary trees. All right, for the rest of our time here, we're going to zoom in on pairwise alignment, because it's built on two of the most famous, most foundational algorithms in all of bioinformatics. You can think of them as the Alignment Detective's toolkit. Two different tools for two very different jobs. So inside that pairwise toolkit, you've got global and local alignment. Global, which uses an algorithm called Needleman-Wunsch, does exactly what it sounds like. It looks at the whole picture. It compares two sequences from beginning to end to end. The analogy here is perfect. It's like comparing two novels to see how similar they are overall. But local alignment, totally different philosophy. It uses the Smith-Waterman algorithm, and it's not trying to compare the whole thing. It's a sniper. Its only job is to find the best matching region inside those two sequences, even if the rest is totally different. It's like finding a single, perfectly shared paragraph in two otherwise unrelated books. So how does this global alignment magic actually work? Well, conceptually, it's pretty neat. The computer builds a grid, right? Puts one sequence along the top, the other down the side.
Then it goes cell by cell, calculating a score. You get points for a match, you lose points for a mismatch or a gap, but the real genius is the traceback. It starts at the bottom right corner and works its way backward, finding the highest scoring path to the top left. And that path, that is the optimal alignment. It's like solving a maze where the path spells out the answer. Now, local alignment uses a similar grid system, but with a brilliant little twist in the traceback. Instead of starting at the end and going to the beginning, it finds the highest scoring cell anywhere in the whole grid, and then it traces back from there, but only until the score drops to 0. And that's it. It stops. This simple change is what lets it perfectly find that one little island of similarity and ignore all the noise around it. And this exact technique is the engine behind one of the most famous tools in biology, BLAST. It's perfect for sifting through massive databases. Okay, let's pull back out to the 30,000-foot view again. We've gone kind of deep into the how, but let's connect it all back to the future of medicine. We started this whole thing talking about a pandemic, but believe me, the implications of this are so much bigger. Because when you boil it all down, when you strip away all the complexity, it really just comes down to this: one single tool. And with that one tool, we're unlocking the future. We're talking about personalized medicine designed just for your DNA. We're talking about finally getting to the bottom of complex genetic diseases. We're talking about therapies that, honestly, we can barely even imagine yet. It all comes from this newfound ability to not just read, but to understand and compare the language of life itself. And you know, this isn't just about what things do, it's also about where they came from. As the quote says, when two sequences align really well, it's a huge clue that they probably share a common ancestor. So we're not just comparing computer code here, we're reading history, our shared biological history written in the language of our genes. So I'll leave you with this thought. We now have the ability to read the book of life. For the first time, we're fluent in its language. So the question for us, for humanity, is no longer if we can read it. The question is, what chapter should we read next?